All right, welcome to the last week of instruction for the neuroanatomy module. You all got the announcement I sent last Friday, right? About the really long-winded one. Yeah, okay. So I hope you took advantage of that advice. I hope you studied this weekend because we're pretty much hitting crunch time, and we're gonna tie a lot of things together in lecture and lab this week. So I'm gonna finish talking about the last sensory system that we'll cover, and that's the olfactory system. And we've already pretty much covered it already. So, but we're just going to go into a little bit more detail. So remember, I drew this diagram on the board on the first in the first lab. I tried to draw it about the part that the first olfactory nerve, the olfactory nerves are going to be like in the holes under the skull. See that? And I mentioned that if you remove the brain, those olfactory nerves or olfactory nerve number 1 get left behind. Like I was trying to kind of draw this. So the first cranial nerve, olfactory nerves, will synapse on the olfactory bulb. So that's the olfactory bulb right there. Specifically, the cells are called the mitral cells. The mitral cells of the olfactory bulb. And these mitral cells of the olfactory bulb, on which the olfactory nerves number 1 synapse on, will give rise to some of the axons that will leave the olfactory bulb. And so this stretch of axons that leave the olfactory bulb, they leave it laterally. And we've already met that track. We've already seen that track in our very first week. Can you kind of remember what it is? These are the olfactory neurons that leave laterally. Any guesses? The name is quite literal. Not lemniscus, but lateral olfactory tract. Yes, that's it. So that's where they come from, right? Remember this? The lateral olfactory tract, the white, where is it? Yeah, see? The white, from bulb, the white streak that goes to the pear, remember? Right? So that's it. That's where they come from. I said you went to the piriform cortex, which is where smell is processed. But there are actually 3 targets of this lateral olfactory tract. There are 3 targets. One of it we already talked about the piriform cortex, the primary olfactory cortex, also known as the piriform cortex, that pear-shaped one. Remember, that's where smell is processed. It has 2 other targets. This lateral olfactory tract. One of it is the amygdala, and the other one is the entorhinal cortex. We have seen amygdala, we saw it last week. Entorhinal cortex, we saw in the first week, right? So entorhinal cortex is a part of that piriform cortex. So that's the 3 targets, the 3 targets of the lateral olfactory tract: piriform cortex, amygdala, as well as entorhinal cortex. Just follow this one over here. So in the sheep brain, we just talked about the lateral olfactory tract. So the olfactory bulbs give rise to axons that leave laterally, lateral olfactory tract into this thing that, if you were to look at it, turn it, flip it that way, it looks like a pear, that piriform cortex. And there's a rhino fissure there, like the one that. What is the region that? The rhino fissure is a border of a region that has a name. What is the name of that region? No, like specifically like here, medial to the rhino fissure, like this region my mouse is covering right here, this whole region. There's a name for that. It sounds like, you remember LA and Westwood? Rhinencephalon, yes, that's it. This part is called the rhinencephalon, which is part of the telencephalon. Remember that? See, this week in lecture and lab, we're going to start tying everything together, right? Okay, so that's pretty much the olfactory, that's one part of the olfactory system, the olfactory pathway. There's another one. This is the lateral olfactory tract, right? The existence of a lateral olfactory tract implies the existence of what? Medial olfactory tract. There is a medial olfactory tract. We just don't see it. It's actually subcorticals underneath, so we won't see it. But we have talked about its connections before. So here is like a stain of a red olfactory bulb. So this is the left and right olfactory bulb of the rat. And you can see here the axons coming out of the olfactory bulb. There's another, so axons go out through the lateral olfactory tract. There's also another one. That's the medial olfactory tract that we won't see, but it exists. And this is one obviously goes differently than the lateral. This one comes here, and then it joins this major axonal freeway that we've met in lab last week, the anterior commissure. Remember the anterior commissure, I said, connects three things, and one of it is both olfactory bulbs. This is how. So it comes here, and it joins the bundle of axons like a freeway, like, joining an on-ramp of a freeway, and then after it crosses the midline, it exits and goes to the other olfactory bulb. So that's why the anterior commissure also connects both olfactory bulbs. See that? Right. So on this side, the medial olfactory tract will come in and join the anterior commissure and then exit to the contralateral olfactory bulb as well. So yeah, it's a very simple pathway, but there's kind of two of them. So the olfactory nerves, number one, will synapse on olfactory bulbs. Olfactory bulbs will go through ipsilaterally through the lateral olfactory tract to project to three places. The piriform cortex on the same side, including the entorhinal cortex as well as the amygdala. Remember that? And the other one will go ipsilaterally through the medial olfactory tract, and then it joins that freeway anterior commissure to go to the contralateral site, and then it exits and goes to the contralateral medial olfactory tract to the contralateral olfactory bulb. Right. So the olfactory system is a very good starting point to talk about our main focus for lecture this week. That is the limbic system. I mentioned last week in lab that we saw a few members of the limbic system, and we're going to talk all about it. And it's here right now. It was originally not discovered, but like one of the first people that kind of thought about the structures that eventually became what we now know as the limbic system, started with James Papes, and he wondered what happened to people who contract rabies. We heard of rabies, right? Like you get bitten by it's a virus, it's a rabies virus, like a wild animals have it, carry it. If you get bitten by a wild animal, you get the rabies virus. The rabies virus will attack you, not just you, but like any creature that's bitten. Now, animals that have rabies, the symptom is that they go through very profound changes in temperament and behavior. They become extremely aggressive. And maybe you've seen movies before of rabid animals, like they foam at the mouths, and it's very dangerous. It is actually fatal if it goes untreated. But there's a vaccine for it. So if you get bitten by a wild animal, better just go get the rabies vaccine just in case. So because the virus caused such profound changes in temperament and behavior, James Papes was one of the first people to wonder what happened, what exactly, which parts of the brain does the rabies virus attack? And he found several implicated structures that eventually known as, well, at first it's called a Papes circuit that has evolved over time to more people studying it, and now today we call it the limbic system. And that's basically the limbic system right there. And this is the diagram that you need to memorize as far as connections of all the structures in the limbic system. And I'm going to talk about each connection one by one, but they're all summarized by this diagram over here. Let's start over here. Olfactory bulb, we just talked about it. The olfactory bulb, one of the targets of the olfactory bulb to the lateral olfactory tract is the amygdala, one of those three. I just talked about that. I had not mentioned this previously, but the olfactory bulb, see here, also projects to the septal nuclei. So olfactory bulb, so amygdala right here. One thing. So one of the structures in this diagram is see this thing called the hypothalamus right here. So the hypothalamus is generally not considered part of the limbic system, but it is a structure that has a lot of connections with the structures that are considered part of the limbic system. And in lab last week, we saw the amygdala. There was a second coronal cut that we did. That's right lateral to the tree trunk, which is the internal capsules right lateral to it. And this, if you look ventrally, this bump over here that we cut through this bump, what is that bump called? We cut through it to find the amygdala. Uncus, right? So the uncus is right here, the ventral part. You saw amygdala over there. Um, and the amygdala here, you can see it will project to the entorhinal. See, follow this red arrow here. Will project to this thing, the entorhinal cortex. So amygdala will also project to the entorhinal cortex. And we saw the entorhinal cortex in the very first lab, the very last part of the first lab, when we talked about what is this entire thing called again? Piriform cortex. Like this region is a landmark called the uncus, and then this one will be the entorhinal cortex, which is a landmark for what? Hippocampus. Remember I mentioned that if the hippocampus is inside there, right? In fact, if you look at this diagram, entorhinal cortex, hippocampus is inside there, right? So yeah, so the entorhinal cortex. And yeah, I also note these two red arrows. There are reciprocal connections, projections between the entorhinal cortex and the hippocampus. Two-way street, okay? The hippocampus is very famous. We talked about it a little bit, but you talked about it in previous classes before. And when we talk about modern studies of hippocampus, I'm sure you've all heard of patient HM, right? Very famous case study. He was 7 years old. He was riding a bike without helmet on. He hit his head, and for the rest of his life, not the rest of his life, until his surgery, he had very horrible seizures. There were hundreds of seizures every day. He was not responsive to medication. So so he passed away in 2008. So then his identity was revealed after he passed away, because he passed away. Henry Molaison, and as a very kind of a drastic way to treat his seizures. His seizures always started from the medial temporal lobes, including the region that included the hippocampus. So since that's where all his seizures started from, easy way to, not easy way, but kind of a drastic way to treat the seizures is to remove that area so that you cut it off at the source, right? So they limited his seizures quite largely. And so he underwent a large medial temporal lobe resection. So like the medial part of the temporal lobes, they were removed, including the hippocampus. And that really helped his epilepsy. But at the time, they didn't know the role of the hippocampus in memory yet. It was him, kind of the first case study that sparked it. He obviously, as you know, had suffered from very, very severe anterior great amnesia. He could no longer form new memories, like commit new memories to long-term memory. So I don't remember what age he got his the surgery, but he basically, as far as his memory is concerned, he's stuck in that age because he can no longer form new memories, right? So there were many people that studied him, very famously William Scoville and Brenda Milner. They studied him for the rest of his life. They kept going to him and asking, that's when they knew about really memory. And memory, there's not just memory, there's explicit and implicit memory, because there are some things that HM could learn going forward. And there was like motor skills, like riding a bike, for example, or like motor things like muscle memory. He could still learn those, but it's just semantic things like explicit memory that he could not remember. So it was because of him that they learned, oh, now we know that there's actually implicit and explicit memory. And because after the surgery, patient HM was still able to learn implicit memory tasks, that implies that the hippocampus is only implicated in explicit memory and not implicit memory. Like that's how it all came about. Yeah, they studied him throughout the rest of his life. William Scoville passed away a long time ago. Brenda Milner is still alive. She is over 100 years old. And in a human scan, so this is an MRI scan of patient HM, and I mentioned last week that everywhere that's black in a MRI scan is fluid-filled spaces. So you can see these two black holes right here. That's where the hippocampus used to be. which is okay. So that looks a bit different than your sheep brain. You remember the sheep brain, the last dissection we did that we saw the really giant, large hippocampus. There was like pretty much like the whole brain's like here and here, right? In that's sheep and other creatures, but in humans, our neocortex, has evolved to expand so much that over evolution, the cortex has gone so big they kept pushing and pushing and pushing and pushing the hippocampus down. So in humans, hippocampus is just right about here. All the cortex has pushed it down through evolutionary time. Right? And that's where the hippocampus used to be. And the hippocampus, the word hippocampus means seahorse. I think I mentioned that already. Greek for seahorse. And the reason why it's called seahorse is because hippocampus, seahorse. It looks like it right there. And this is a human, a diagram of a human hippocampus. And this ties back to the video that I showed last week, the purple banana, right? Purple banana coming out here, giving rise to axons called the fornix, and then terminates here. One of the branches that go rostral is the one that goes rostral. Septonuclei. Yeah, that's what this diagram is depicting right here. So yeah, hippocampus. This is the last slide that we did in lab last week. These two giant things on either side. There's a hippocampus. More recent studies of the hippocampus have revealed that it has a more specific role than just consolidating short-term memory to long-term memory to the cortex. It's very involved in spatial memory, especially like memory of space. So they found in the hippocampus, you may have heard of this from other courses, these cells called place cells. So these place cells in the hippocampus are sensitive to locations, essentially. So for example, this is kind of from a very well-known paper on place cells from O'Keefe and Burgess in 1995. So they placed electrodes into a rat's hippocampus to record these place cells. So they record whenever it fires, the computer will capture whenever it fires, right? And they put a rat in a square arena and just allow the rat to just roam around there. And everywhere, so this diagram up here tracks the path of the rat. So the line here, the line is the journey, is the path of the rat. And everywhere that the place cell fired, each dot here is everywhere that particular cell that it was being recorded fired. So you can see here that the cell seems to prefer firing only when the rat is around this region of the arena. So if you average it out and took a heat map of it, this is what it will look like. So we call this a place field. So that particular place cell that was being recorded has a preferred place field of this location right here. It seems to only fire when it's there and not anywhere else, right? And different place cells have different place fields. So here is a recording of 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 30. 30 different place cells, and each of them have a slightly different place field, like where they prefer to fire. See that? Right. So every cell has a different place field. So together, it tiles up in the representation of the spatial environment, and evidence that the place cells are responsible for spatial memory. You may have heard of other experiments before, like the Morris water maze. Like if you get a rat to swim in water to a platform, like a hidden platform, like the water is all milky, so the rat can't see. But there's a hidden platform, and you just put a rat in that pool, the rat will swim until it finds that platform. And then you take it out, you put it back again. And each time, the time taken for the rat to go to the platform is smaller and smaller because it's learned where that platform is, right? If you lesion the hippocampus, you put it back in the water, it's like as if it's never learned. It's like the first time, like your spatial memory. Right. So that's hippocampus, very well known for that. Okay, this part we saw in a video. So this is the purple banana, the hippocampus. Gives rise to axons, and we just talked about it. It will form the axons called the fornix. It will arch rostrally, and then it will come over here where it splits. It is not shown in this diagram over here, but there's something here that where it meets where it splits. Where's that thing should be right here, where it splits. Anterior commissure, very good. So it should be like right about here, but here's where it splits. One branch that goes rostral, we just talked about the septonuclei. And we technically saw saw it in the first dissection. Here, the fornix, let's see this right here. There's a fornix. This is that top of the arch. That's this equals this. So that's part of fornix. We'll come over here. We'll hit the interior commissure where it splits. And this is, well, in the first lab, we saw this. We talked about this in the first mid-sagittal dissection. I had septonuclei in parentheses because remember in the mid-sagittal view, it's not the best view of the septonuclei. It's right there. It doesn't have a shape or anything like that. But in the first coronal cut that we did last week, we saw it right there. This thing right in the middle. And then in the second coronal cut, we saw a bit of the fornix. Here, there's a fornix. This is the second coronal cut that we did. This is the slice that we saw. Do I see it here? Actually, no. This is a slice that we saw that we saw like fornix is like this is actually fornix over here as well. No. It's a horrible diagram, but there should be 4 white dots. 1, 2, 3, 4. It's not here, but we saw the 4 white dots. 1, 2, 3, 4. And the 2 one here is fornix. We saw the fornix twice because the fornix is a C-shaped thing, right? And you cut through it, you see it twice, right? And then, okay, so one branch will split off and terminate in the septonuclei. Look at this arrow here. Remember that this is that two-way street. Septonuclei projects back to the hippocampus, also to the fornix. The other one is only a one-way street, right? It will come over here. After it gets split, the part that goes caudal and ventral to the mammillary bodies right here. And that's a one-way street. And we, in a mid-sagittal view, that's the mammillary bodies right there. That's the optic chiasm. What is this round thing? What's the circle? Thalamus, right? So if this circle is the thalamus, this region is the hypothalamus, right? The mammillary bodies are part of the hypothalamus right here. In a ventral view, we saw that right here. So in the ventral view, this is the last thing that we did in the first lab. The first lab we didn't do any cuts, right? We only saw the ventral part, and all that we can see of the diencephalon is just this little region right here, and that's all hypothalamus right here. But then this particular part is the mammillary bodies right here. We talked about what happens if the mammillary bodies get destroyed, but remember that the guy that broke in a bar fight, remember what happened to him? Very severe anterograde amnesia. There was another way that that the mammillary bodies could get damaged, not through physical damage, and that is known as Korsakoff syndrome right here. So here you can see that's the left and right mammillary bodies, and you can see here the left and right hand mammillary bodies over here. Where you see black is where the cells are died. The cells died and became scar tissue essentially, like the corpses of the neurons. There's all the black stuff right there, and that's called Korsakoff syndrome. Korsakoff syndrome is once upon a time we thought it was caused by chronic alcoholism because we see it a lot in chronic alcoholics. So we thought that one long-term effect of chronic alcoholism is the destruction of the death of these mammillary bodies. But later on, we figured it's not quite alcohol per se. It's because of in long-term alcoholics, the alcohol interferes with their body's ability to absorb thiamine, also known as vitamin B1. So thiamine is also known as vitamin B1. So long-term, alcoholism interferes with the body's ability to absorb vitamin B1, also known as thiamine, and that results in the death of the mammillary body's cells. So yeah, so these are examples of Korsakoff syndrome. This is the mammillary bodies over here. And then the next connection, this is where the video with the purple banana kind of stopped. And this part wasn't on the video, but I was using my fingers to try to draw on the screen, right? Okay, when I drew it, it was parallel, but this is another version of it. But there's axons that go up here to the anterior nucleus of the thalamus. Remember that? right? I know the shape here looks different than in sheep brain, but this is human, so this is a bit different than sheep. In sheep, this track goes parallel, if you remember, um, and okay. So if this track of exons goes from the mammillary bodies to the anterior nucleus of the thalamus, the name of this track is mammillothalamic tract, right? And yeah, okay, so this is what, oh, we do see four dots. I can't see it from here, but like one, two, three, and I guess four. Yeah, there we go. One, two, and three, four. The two dorsal ones are the mammillothalamic tract we saw that in the second coronal cut that we did. The mammillothalamic tract is always right next to the thalamus. We're going to be doing, we're going to do one more coronal cut in lab this week, and then we're going to do two, because we haven't done horizontal yet. So in lab this week, we're going to do horizontal cuts. And horizontal cuts, you will see, again, four dots. And four dots, they are the very same four dots. Two of them are going to be the fornix, two of them are going to be the mammillothalamic tract. And in both cases, the mammillothalamic tract is always right next to the thalamus. So there's one easy way to just tell it apart. But don't worry about the horizontal yet. We'll get there when we get there in lab. I'll remind you of this. Okay, and then I did not, in lab, when I talk about, the projections from the mammillary bodies to the anterior nucleus of the thalamus, I just stop right there. It does go on, and that's represented here by those huge arrows here. The anterior nucleus of the thalamus will project to the cingulate gyrus or cingulate cortex that we saw also in lab last week in the mid-sagittal view. The giant gyrus is kind of sitting right on top of the corpus callosum. Right here. So that's the corpus callosum right here. There's a cingulate gyrus right here. There's gyrus right on top of it, cingulate gyrus or cingulate cortex. So that's where the anterior nucleus of the thalamus will project to. So yeah, I talked about the cingulate gyrus being a part of the limbic system. Here it is. And finally, so the limbic system is kind of like a loop. The final connection that makes it a loop is this arrow right here. The cingulate cortex projects to the entorhinal cortex. The cingulate cortex or the cingulate gyrus projects to the entorhinal cortex right here. Oh, I forgot one thing early on. When we were talking about amygdala, I said I talked about the amygdala projecting to the entorhinal cortex, right? I forgot this. See this? Trail of axons from amygdala to, just follow it, to the hypothalamus. Excuse me. The amygdala projects to the hypothalamus. And the trail of axons, well, everything has a name. Every trail of axons has a name. And the name for this one is the stria terminalis. The amygdala projects through the stria terminalis to the hypothalamus. Okay. So as far as the limbic system goes, here's the summary. So these are the members of the limbic system, and these are the tracks that connect things, axonal tracks. So in the cell bodies, we have the amygdala, the hippocampus, entorhinal cortex, septal nuclei, mammillary bodies, anterior nucleus of the thalamus, and the cingulate gyrus. And the axonal tracks with stria terminalis. Stria terminalis connects we just talked about it, the amygdala to the hypothalamus. And here's one thing that you should be careful of. I see it quite frequently. Students confuse hypothalamus with hippocampus. They're both long H words, but they're very, very different things, right? So don't fall for it, right? Keep hippocampus and hypothalamus straight, right? which brings me to a question that a few people have already asked, and I get students asking me all this every time. Spelling, does it matter on the exam? So yes, just do your best, because if you don't spell well, we cannot grade what we don't understand. So just do your best. We can forgive minor spelling errors, like if you spell mammillary bodies with an M instead of two. Yeah, that's okay. But and this has happened before. Like students will try to disguise, try to fake spelling errors. Like they are not sure if the answer is hippocampus or hypothalamus, so they put hypothalamus or hippocampus. No such thing. So that you're just fishing. You're just fishing there, right? So that's not a spelling error. That's a crime. So don't do that. Okay. So just do your best when it comes to spelling. Yeah. So the stria terminalis connects the amygdala to the hypothalamus. The fornix connects between hippocampus to septal nuclei, two-way street, and hippocampus to mammillary bodies, one way, right? The mammillothalamic tract, very literal, mammillary bodies to the anterior nucleus of the thalamus. I included this slide accidentally, just ignore it. I don't know. This was from last quarter. It was relevant for something last quarter, but not this quarter, so ignore that. So yeah, as far as lecture material, you'll be glad to know that this is it for lecture. As far as lecture material, I mean, obviously, it's going to be connected to lab, because we're going to see all these connections and all this stuff in lab. We're going to talk all about that. But I'm gonna spend a bit of time to actually give a little bit of a revision, if you will, and if also to kind of see what you've been doing this weekend. So this is a diagram of basically the corticospinal tract, which is what function? Motor, the one and only major motor system that we talked about, right? Corticospinal tract. Where's it start from? Precentral gyrus, right? So yep, there in the precentral gyrus. There's a somatotypic representation of it. So the muscles of the finger are represented by the neurons over here, and so on and so forth, in the face. And all those axons will come down and bundle, and as it kind of goes down, it will form this thing that in the coronal slice looks like a tree trunk. What is a tree trunk? Internal capsule, which are axons, right? So internal capsule are axons going up and down, but it looks like tree trunk always. All right. And then it's going to go down, 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 down. As it passes through the mesencephalon, it's going to form what? The cerebral peduncles, good. And then the axons will continue going down, 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 through kind of underneath the pons, and then goes down, 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 and I guess it's not shown here, but in the myelencephalon, it's going to form these two columns called the pyramids. And then in the sheep brain, and also in human, we can see a little bump over here. What's happening at the bump? The decussation of pyramids, right? So I don't know if everyone got the tour. I don't know if this was mentioned on the tour, but on the exam, we're going to be very specific. So if you see this part of the sheep brain, and if the pin is specifically pointed on the bump, if the pin is on the bump, we are talking specifically about the decussation of the pyramids. If the pin is just up here before the bump, it's just pyramids. Right, so the placement. Okay, yeah, I mentioned that the practice exam is now available, right? So you could print it and bring it to lab, and you'll see. So you'll be doing the practice exam. The practice exam was last quarter's exam. So our slices will have pins in it. The placement of the pins are very deliberate. There's always a reason why the pins are where they are, right? So it decussates, and okay, so let's finish the pathway. After it decussates, you'll go down and terminate where? I can hear you. It sounded correct. Correct. The lower motor neurons, which live in what? The ventral horn of the spinal cord. Yeah, right here. The lower motor neurons in the ventral horn of the spinal cord. And then where will the lower motor neurons in the ventral horn of the spinal cord project to? Muscles. Good. That's how we move. What happens if the upper motor neurons get damaged? Spastic paralysis, contralateral side, right? What about damage to lower motor neurons? Spastic paralysis, ipsilateral side. Okay. Here's one thing that you'll get to as you do the practice exam. Will tell you. So read the instructions. So on the first page of the practice exam, the instructions, they're going to be the same as your exam. Read it carefully. On the exam, we don't want you to use the word ipsilateral or contralateral. We want you to specify left or right because this is an anatomy class. I want you to show me that you know which is left and which is right. Right. So don't use contralateral, ipsilateral in exams. Use left or right, specify it. Also, do not use the words motor cortex or somatosensory cortex. Because I want, again, this is an anatomy class. I want you to tell me that you know where it is, right? So instead of motor cortex, it should be pre-central gyrus. Instead of somatosensory cortex, it should be post-central gyrus. Good. All right, what else can we talk about here? Oh, okay. This is the mesocephaline. You'll go through the two cerebral peduncles. When you get through the cerebral peduncles, you should expect to see these cells here that are colored. They have a specific color. What are those? Substantia nigra, that's where it should be. In humans, it's really black. That's why it's called substantia nigra. But in sheep, there's no color. But there's only one coronal cut that you see. There's only one cut, period, in the whole module where we talked about the substantia nigra. So if you see that slice, that's the only slice we could ask about it on. But again, as you do the practice exam, you'll get more of a hang of it. Yeah.
Go over like the specific like start to end of the limbic system.
The limbic system is just basically it's just this diagram over here. That's why I said this is a summary of everything we talk about. Just start. It's a loop, so it really doesn't start anywhere. So we can say arbitrarily we can start from olfactory bulb,
So
right?
like to the central nuclei, is that a whole different pathway? Because that's different from like the amygdala. That make like after the olfactory bulb, it goes straight to the amygdala. Okay, so it doesn't go to the central ventral.
It does. It also does.
Okay, so
Yeah.
like how do we know?
It happens at the same time.
Oh,
Yeah. So it's not like number one, number two, number three. It's all connected and all happening at the same time. So it's hard to answer like, where does it start? Where does
they
it?
both go to the hippocampus.
Yes. Yeah. So there are a lot of common projections in, there's quite a bit of redundancy. So definitely spend some time really, really soaking in and following the arrows in this diagram to memorize the limbic system, definitely. Right. Okay. Yeah.
How in depth do we make each know about the limbic system's functions?
This, like the
Or
connections,
So like arousal, fight or flight.
whatever we talked about.
Oh,
There's so much that we did not talk about, but whatever we talked about, like, hippocampus, memory, amygdala, fear. Well, we didn't talk about fear, but you all know from other classes, right? Amygdala, fear, which is an emotion, right? Subnuclei emotion, like, if you damage it, you get separate rage. We talk about that. If you stimulate it, it feels really good, right? Like things like that, right? If you lose your hippocampus, get memory problems, especially spatial memory, you will lose, you'll be lost essentially, like spatial memory. All right, in this diagram, I can tie it to something we talked about. So in the mesocephalon, what are these two bumps here? Cerebral peduncles, right? And can you see here, there's a cavity in between that's going to be filled with fluid in a real living creature. Everything has a name. That fluid-filled cavity also has a name, and that name is the interpeduncular cistern because it's right in between the two cerebral peduncles. And then you can see here, the corticospinal tract comes down, and then a part of it synapses in a pons. See that? Right? And then the pons will then project to where? Cerebellar what? Specific. Which part of cerebellum? Cerebellar cortex. Yes, cerebellar cortex. And the bundle of axons that goes from the pons to the cerebellar cortex is middle cerebellar peduncle, right? Yes, and that carries the copy of that motor cortical spinal motor information to the cerebellum. That's how it does its job. So like things like this is how you should be studying. Like I mentioned in that long-winded email, like group study is honestly the best way to go. Like look at the picture. Okay, what else can we see here? Like even if the picture, like we talk about the oh well, let's talk before that. So that one that goes from up there to this one to the cerebellum, there's a name for that track. What is that name of that track? Cortical ponto cerebellar tract. Like yeah, things like this. Like this is how you should be studying with when when you get together in groups. Even if this diagram is just talking about cortical spinal tract, but there are other things that we can see here that relate to other things that are relevant as well, right? So like quiz each other. Like what is this? What is that? What else can we see here? And this doesn't show here, but after that, where does it go? What does it do? Like that's how you should be studying, like quizzing each other really over and over and over again, because again, it's all about memorization, and memorization requires repetition, right? And not and you learn. I hope you have learned about the difference between mass learning and space learning before, right? Like if you cram, you are more likely to have interference of the memories that you form. But if you space it apart, then the more likely it's going to be more solid, and you won't forget it. You won't confuse the other things. So don't study last minute. Space it apart. Like you have a whole week, a little bit more than a week between now and your exam. So we got to be doing that regularly. Uh, so yeah, I think that's what is the name of this cranial nerve that runs this way? Spinal accessory number 11, right? Yeah, things like that. Just whatever you see, just ask questions. Remember cranial nerves, give both name and number for full credit, right? Remember, don't just give name alone or number alone when you're asked to identify a cranial nerve. Give both for full credit. Okay. So when we get to the first part of this week's lab will be finishing the sections with one more chrono to go, and then we'll be doing 2 horizontal cuts, and after that we'll give you time to with the practice exam, and that's where you'll kind of get a hang of how the exam is going to be. It's going to be stations, like there's going to be, as you see on the exam, that there are stations. There are 25 stations, which means there are 25 slices. Each slice has 4 questions. Each station has 4 questions, like with pins in it. Everyone who's gonna be is gonna be at one station, and you're just gonna be going around. So I mean, ever been to one of those sushi restaurants with a rotating sushi thing, right? It's like that, but you order sushi. You're gonna be rotating around the stations, and in the first round, everyone's gonna get 2 minutes, 2 minutes per station. And then there's going to be a timer. Timer is going to beep, and then you're going to move to the next station. 2 minutes, beep, move to the next station, right? So everyone's going to do that. After everyone has been through the station at least once, no, everyone exactly once, after everyone's gone to all 25 stations, you can keep going, but then now the time is going to be 1 minute. So 1 minute per station, beep, move. 1 minute per station, beep, go, right? And I know 1 minute sounds like a short period of time, but trust me, when you're there, it's going to feel like an eternity. It's really not short at all once you're there. Trust me. And then yeah, so you keep going around. You'll begin about 2 hours. The exam can be finished in 1 hour. The exam can be finished in 50 minutes, but you'll be given 2 hours. And then what else? Yeah, I mean, if anything that I forget, I can just remind you all in lab. Bring a pen. The final answers must be in ink. You can use pencil to do scratch work, but final answers must be in ink. Yeah, anything that I left out, I will talk about it in lab. Okay. All right. I'll see you in lab. Oh, thank you. You do that now. You haven't taken an exam yet.